This Li-Fraumeni Syndrome iPSC-based osteosarcoma model can help answer key questions in osteosarcoma research field about using patient iPS cells in cancer modeling.
The main advantage of this model is that it allows the generation of unlimited cells at all stages of osteosarcoma development for mechanistic studies, biomarker identification, or drug screening.
Ruoji will be demonstrating the cell culture procedure, and I will be demonstrating the animal procedure.
After 2 weeks of mouse embryonic fibroblast or MEF co-culture, wash the 80 to 90% confluent iPS cell culture with 5 milliliters of DPBS, followed by the addition of 1 milliliter of cell detachment solution to the cells for a 3-minute incubation at room temperature.
It is critical to maintain iPS cells in good conditions on MAFs for at least 14 days for the cells to be able to attach to a gelatin-coated plate for their differentiation into mesenchymal stem cells.
When the cells have detached from the plate bottom, add 9 milliliters of MEF MSC culture medium to the cells to neutralize the cell detachment solution, and transfer the cell suspension into an uncoated 100 millimeter tissue culture plate. Incubate the cells at room temperature for 30 minutes to allow the MEFs to attach to the plate, and carefully collect the unattached iPS cell-containing supernatant. Centrifuge the iPS cell solution and resuspend the pellet in 6 milliliters of MSC differentiation medium.